Hi friends, welcome to EZI Nurse where nursing education made easy. Today we are going to discuss some important exam oriented question from genetics. This is very important. So genetics is important and you are going to write exams for genetics. So yes, I know that you are preparing for your exam. This will help you to prepare more. So this video will help you to get pass in the exam. So focus on this video. So first we are going to discuss about genetics. What is the definition of genetics? So what is the genetics definition? It's a science of heredity and variations in living organisms. So it's a study or it's a science of heredity as well as variations in living organisms. So that is what called genetics. So this is the definition of genetics. Definition of genetics will come for a 2-mark or a 1-mark question. So focus genetics. So now we are going to discuss about another important question that will definitely come for the exam is cell division. Cell division, it's divided into two types. It's in meiosis and mitosis. So let's see what is meiosis and mitosis. So mitosis both in somatic and reproductive tissue. So mitosis, the process of mitosis or cell division is happening inside the somatic and reproductive tissue and two daughter cells are formed. So there is two daughter cells. Genetic uniformity is maintained. So the daughter cells will be having a genetic uniformity. No pairing between homologous chromosome. Each chromosome contains two sister chromatids and centromere divides. So this is the qualities of mitosis. These are the characteristics of mitosis. Now we will discuss about meiosis. What are the characteristics? Reproductive tissue only. So it's a reproductive tissue for daughter cells. So instead of 2 here, 4 daughter cells will form and genetic variability is generated. Pairing between homologous chromosomes. Each chromosome contains 4 sister chromatids and no division of centromere. So this is the difference between cell division process, mitosis and meiosis. So your questions will be, it's coming difference between meiosis and mitosis. So this is the difference. Yes, now we are moving to another important question, deoxyribonucleic acid or DNA. Deoxyribonucleic acid. What is DNA? So DNA is a chemical component of chromosome located in nucleus of every cell. So DNA is a chemical component in the chromosomes and it's located in every cell. Structures of DNA codes for genes. So DNA structures. These are the structures of DNA code for genes. So this is the structures where it code for genes. And the structure of DNA was established by James Watson and Francis Crick in 1953. So who first introduced DNA codes, that is Francis Crick and James Watson. DNA is a double-stranded helix. So you can see here in the picture, it's a double-stranded helix. So DNA is a double-stranded helix and with nucleotides on each strand physically matched to each other. The molecular basis of gene is deoxyribonucleic acid. DNA. So molecular basis is called deoxyribonucleic acid. DNA is composed of 4 main types of chains. What are the main types? Adenine, cytosine, guanine and thymine. So adenine, cytosine, guanine and thymine. So we can see the base pairs, these are the base pairs of DNA and this is the sugar phosphate bond. So this is connected with sugar phosphate bond and the base pairs is adenine, thymine, guanine and cytosine. So this is the deoxyribonucleic acid DNA. Next, we are moving to Mendelian inheritance. So, father, who is Mendelian? Who is Gregor Mendel? He is the father of genetics. So, Gregor Mendel and Mendel's law is law of segregation and law of independent assortment. So, two laws, law of segregation and law of independent assortment. So, what are Mendel's law? Let's divide. Genetic characters are controlled by unit factors and exist in pair of individual organisms. So, according to his law, genetic characters is in unit factors and in existing pairs. When two unlike factors responsible for single character are present in single individual, one factor is dominant to other, which is called recessive. So, one dominant factor will be there and one recessive factor will be there. And during the formation of gametes, the paired unit factors separate or segregate randomly to each gamete received on one or other unit factor with equal likelihood. Two independent characters segregate independently each other. So, this is the main Mendel's law in the case of genetics. So, he is the father of genetics, Gregor Mendel. Yes, now we are moving to another important question, erythroblastosis fetalis. So, what is erythroblastosis fetalis? Let's discuss. So, Rh negative mother carrying Rh positive fetus. So, here the mother is Rh negative and she is carrying a baby of Rh positive. Fetal blood crosses the placental barrier and reaches mother bloodstream. So, from the fetus blood it is crossing the crossing the placental barrier and it enters in the mother's bloodstream. Then the mother forms antibodies against the fetal blood. So, mother's body will start to produce antibodies against that fetal blood. These antibodies return to fetal circulation, which causes damage to fetal blood cells. So, this produced antibody will return to the fetus blood in fetus blood, and there is a damage to fetus blood cells that is fatal to fetus. So, it will kill the fetus. So, that's the erythroblastosis fetalis. So, the Rh negative mother, Rh positive baby. So, the Rh positive baby's blood cells will cross the placenta and enters into mother's bloodstream. So, the antibodies in mother's body, antibody starts to form against this Rh positive blood group. So, that is going again return back into the fetus and it will damage the fetal blood cells. That condition is called erythroblastosis fetalis. Yes, it's very important question. Next one is consanguinity. So, what is consanguinity? It refers to the property of being from the same genetic lineage as another person, or consanguinity is the quality of being descended from the same ancestor as the person. So, it's a group of people that coming from the same genetic lineage. Consanguineous marriages are those between close blood relatives that are normally morally permissible but do occur in some communities between the uncle and niece or between the cousins. So, consanguinity marriages, that means the relationship or the marriage between the close relatives. Example, uncle or niece or with the between the cousins. So, that is now it's medically prohibited because the babies they will have genetic diseases. So, this is consanguinity. Consanguinity refers to property of being from the same genetic lineage. Yes. Now, we are moving to another important question, atopy. What is atopy? Atopy refers to allergic reactions characterized by the action of IgE antibodies and genetic predisposition to allergic reaction. So, atopy is nothing but allergic reactions. So, it's a allergic reaction, that is what called atopy. Atopy refers to allergic reactions characterized by the action of IgE antibodies and a genetic predisposition to allergic reactions. Example of atopic disorders are anaphylaxis, hay fever, asthma, contact dermatitis, urticaria, gastrointestinal allergy. So, atopy is nothing but allergic reactions and there will be the action of IgE antibodies. Yes. Yes, this is also very important. You need to remember these points. Next, we are moving to another important one is hydrops fetalis. What is hydrops fetalis? It's a condition in the fetus characterized by an accumulation of fluid or edema in at least 2 fetal compartments. So, it is a condition in the fetus and there is accumulation of fluid or formation of edema inside the at least 2 fetal compartments. Hydrops is classified as immune or non-immune. So, either it will be immune or it will be non-immune. Immune causes such as Rh incompatibility between mother and the fetus are uncommon. So, it is in immune causes as Rh incompatibility between the mother and the baby, and non-immune causes include congenital infection, twin pregnancy, chromosomal abnormalities. So, this is the hydrops fetalis. Is a condition of fetus is characterized by accumulation or fluid in the at least 2 compartments of fetus. Yes. Now we will move to another important question, spontaneous abortion. So, what is spontaneous abortion? It's unintended termination of pregnancy at any time before the fetus have attained viability. So, means 20 weeks of gestation or fetal weight more than 500. So, the unintended termination of pregnancy before the period of viability, that is what called spontaneous abortion. So, that's also one genetic condition. It's also related to genetics. Next one, neural tube defects. What is neural tube defects? Are birth defect of brain, spine or spinal cord that occurs in fetus within the first month of pregnancy. So, in the first month of pregnancy, we are detecting that there is a defect in brain or spinal cord that in the fetus. That is what called neural tube defects. Another important question, spina bifida. What it is? It's a defective closure of posterior vertebral column. So, there will be an opening in the posterior vertebral column. A defective closure will be there. It may not be open, but spina bifida occulta. If it is not open, then its condition is termed as spina bifida occulta. So, there will be a defective closure of posterior vertebral column. So, yes, this is very important. Hydrops fetalis, spontaneous abortion, neural tube defects and spina bifida is also very important. It will come for a 2 marks. So, 2 marks question it will definitely ask, or either it will ask for MCQ questions. Yes. We will continue Down syndrome or trisomy 21. So that's also very important, Down syndrome or trisomy 21. It's a genetic disorder caused by the presence of an extra 21st chromosome. So there is a presence of an extra pair of chromosome, that is 21st chromosome. Extra pair will be there, that is trisomy or Down syndrome. And another important question we are going to discuss is a test that used to identify genetic disorders, that is our genotyping and phenotyping. So what is genotyping? It's the process of determination of differences in genetic makeup of an individual by examining their DNA sequence. So by examining the DNA sequence, we can identify differences in the genetic makeup of an individual. So that test is called genotyping. Next one is phenotyping. What it is? It's a process of observing and measuring an organism's physical and biochemical traits as the result from interaction of genetic makeup with the environment. So here we are observing and measures an organism's physical and biochemical traits. That test is known as phenotyping. So genotyping and phenotyping is a very important genetic test. Now the next important test used for in genetics is amniocentesis. Amniocentesis. What is amniocentesis? It's an invasive procedure in which needle is passed through mother's lower abdomen into amniotic cavity inside the uterus. So it's an invasive procedure where a needle is inserted in the lower abdomen through inside to the amniotic fluid. Amniocentesis are performed between 14 to 20 weeks of gestation. It's a medical procedure used primarily in prenatal diagnosis of to assess fetal health and detect genetic abnormalities. It is mainly used to assess the abnormal genetic mutation or any abnormal defect in the genetic abnormalities to assess the fetal health. In India, it's only through registered hospitals we can able to see amniocentesis because of female feticide. So after amniocentesis, the baby, we can identify the gender of the baby, and after that, if it's a female baby, in some parts of India, they're used to abort that baby. So due to this, amniocentesis is only done through various hospitals with registered purpose. Yes. Now we will move to another important test, that is chorionic villus sampling or CVS. CVS is a prenatal diagnostic test used to detect chromosomal abnormalities and certain genetic disorders in developing fetus. So it's mainly used to assess the chromosomal abnormalities. CVS involves collecting a small sample of cells from placenta where it attaches to the wall of uterus. So this is the chorionic villus sampling. Now we are moving to another important disease condition that occurs in genetics, that is phenylketonuria. So it's also important 2-mark question. What is phenylketonuria? It's a rare inherited metabolic disorder characterized by inability to break down the amino acid phenylalanine due to deficiency in enzyme phenylalanine hydroxylase. So there is a deficiency in phenylalanine hydroxylase. Due to that, there is inability to break down the amino acid phenylalanine. So this is a rare inherited metabolic disorder. So what is the characteristic? Inability to break down the amino acid phenylalanine due to deficiency of enzyme phenylalanine hydrolase. So this is the definition of phenylketonuria. It's also very important. Next important question that you may come for your next exam, that is maple syrup urine disease. So it's an inherited metabolic disorder characterized by body's inability to break down the certain amino acid. So if that person's body is unable to break down the amino acids like leucine, isoleucine and valine. This inability is due to deficiency in the enzyme complexes branched chain alpha keto acid dehydrogenase. So there is a deficiency in the branched chain alpha keto acid dehydrogenase. This maple syrup urine disease occurs. Yes. Now we will move to Human Genome Project, HGP. So it's an international collaboration that successfully determined, sorted and rendered publicly available resources of almost all genetic content of chromosome of the human origin, otherwise known as human genome. So it's a determination, sorting and rendering of public available sequence of human genome. HGP had started in 1990 and completed in 2003. It's a 13-year project. So HGP is Human Genome Project. It's also very important. So don't forget while studying, focus. Yes, now we are moving to gene therapy. Important treatment that present in the genetics is gene therapy. It's a technique used to correcting the defective gene responsible for disease development, that is gene therapy. Steps in gene therapy: identification of defective gene. First, identify which gene has the defect. Identification of target tissue and organ, which organ is affected. Packing of normal human genes in suitable vectors. So, next procedure is packing of normal human genes in suitable vectors. Introduction of genes in target tissue or organ. Observe for desired result and monitor for side effects. So, these are the main steps in gene therapy. Now we will see cloning. Cloning is the process of making identical copies of an organism, cell or DNA sequence. Like we are taking a copy. So how we are taking? Cloning is the process of making identical copies. We are making identical copies, exactly the same copies of an organism, cell or DNA sequence. Molecular cloning is a process of which scientists amplify a desired DNA sequence. So if scientists want a desired sequence, they use molecular cloning. Cloning is also asexual reproduction. So it's coming under the category of asexual reproduction. Now we will move to genetic counseling. As a role of us in genetics, it's genetic counseling, communication process which deals with human problem associated with the occurrence or risk of genetic disorder in a family. So it's basically about the genetic disorders, and its management. That is what called genetic counseling. Next one, legal and ethical issues, role of nurse in genetics. What are the role of nurse? Taking informed consent. We need to take the informed consent. Assure privacy and confidentiality. Respect and support counsel's decisions. What the decision? Respect and support the counsel's decision. Like the respect the counsel's means the parent's decision, and providing unbiased information about diagnosis, treatment and disease management to counselor or family members. So, give the unbiased information to the family and the counselee. And genetic specialists and nurses participate in genetic counseling make every attempt to respect the patient's ability to make autonomous decisions, respect the patient's ability to make the decisions, and confidentiality of genetic information and respect for privacy are other essential principles underlying genetic counseling. So, keep the confidentiality of the information that the counselee is providing to the counselor. So, these are the main role of nurse. Till now, we had discussed some important exam-oriented questions from genetics. These questions will definitely come for the next exam. Study well and prepare well. All the best for your exam. For more videos, do follow EZI. Thank you.